Welcome, everyone. Today's our March version of the Statistics Former Students Network webinar, and we're very fortunate to have with us today one of my former colleagues, Auston Way. Auston is a very experienced biostatistician working in the cancer field, and he's going to share some of his experiences with us. He's currently a principal biostatistician team lead at the Lerner Research Institute at the Cleveland Clinic. And he's also the founder of his own business, Waystats. He holds master's degrees in both statistics and biology from Texas A&M University and has extensive experience in biostatistics, statistical programming, and clinical trial design. With a career spanning more than 2 decades, in the roles at the University of Texas MD Anderson Cancer Center and the Cleveland Clinic, he's contributed to numerous high-impact medical and statistical research projects. His expertise includes categorical data analysis, longitudinal data analysis, survival analysis, and he's also been recognized with multiple awards for his contributions to statistical and medical research. So welcome, Auston, and please take the floor.
Well, thank you so much. Well, it's kind of thrilled to be invited by Marcy to this talk, and I wasn't sure what to talk in the first, and then I think maybe as a master level statistician, it's good to share some experiences, my own experiences in the career, and first some update of the my background now. It's I'm part-time working at this Cleveland Clinic and full-time consultant serving Talperion. So this is where the slideshow. I don't want to use CCF logo because I don't want to get into trouble. So I asked the Talperion, the CEO, Rick Landon, and he said, well, sure, you use Talperion logos and share the information of our company. And Rick is in NAGI. He graduated in '92. With a doctorate degree, worked with Dr. Carroll. And he's our CEO, and the CPO, Chief Product Officer, Stephen Eckert, is also NAGI. He graduated in '95. He also won 93 Connor Awards in our department. So Talperion is like deeply connected with Texas A&M Stat Department. All right, so this is, let me see. Okay, so that's how, well, howdy. I haven't said this word for a long time. I still remember when I first came on campus in 1999. It's a hot summer day, and I saw a cadet senior wearing senior boots on campus. And I was like, wow, that boots looks so cool and also so hot on a summer day. So, and when I met his eyesight and he yelled howdy to me, I was like, oh, wow. I was startled a little bit, but yeah, howdy is the way we greet each other. And you know what? I wear boots to work every day now, even in summer day. So I don't want to be kind of, this will be a very informal kind of talk and discussion. The experiences I have during my career, and I want to go through some case studies or more like key functions that I kind of perform on a daily basis as a master level statistician. So to me, there's 3 kind of most important attributes or things that kind of I want to hold on to. One is service, that's a science, and also business. So we would need a service attitude to engage with the clients or collaborators, and we need to learn the science in both statistics and biology and clinical cancer in this. In my specific case, and also we will have to work with a business mind that is thorough and detailed and reproducible. All right. So let's start with my very first interview at MD Anderson. So Marcy, Mark, and Joe, they were all Aggies and who worked at MD Anderson. They all interviewed me when I applied for MD Anderson. And Mark asked this question when I interviewed with him. Suppose I'm a cancer doctor, please explain what are type 1 and type 2 errors. And at that time, I didn't know much about clinical knowledge. So I used the apple and an orange as examples. If you want an apple and someone gave you an orange, you said that's an apple, and that's a type 1 error. And if you, on the other way, the other way around, as a type 2 error. And he said, that's pretty good, but not exactly what we want for a biostatistician working with a cancer doctor. So I learned from that we need to understand first the statistics. That's the science part. What the terms mean in statistics, and then what the terms translate to clinical service, like to doctors, and how to effectively communicate with doctors. So that's the first lesson I learned when I get interviewed. That's how these three things will make the biostatistician work useful or helpful. So after I got into MD Anderson and start protocol review, and that's still a kind of standard workload of statistician, both the master level and the PhD level. So we review clinical trials. MD Anderson is run a lot of clinical trials, and all the protocols has to be reviewed by a stat department. And we make sure the consistencies between objectives, endpoints, and the design. And we go to protocol review meetings. So I personally learn a lot reviewing protocols and going to the meetings, both from the protocols and also from colleagues discussing the protocols, how to provide advices for alternative designs, a better design, how to make a study more rigorous, and also how to kind of phrase the concerns, the feedbacks to the sponsor. So that's kind of cover all the science, surveys, and business part. And this is a great starting point for me, at least. Another thing we usually do is clinical trial design. But here, one example here is not for clinical trials specifically, but for animal study design. It's ACUF. I don't remember what does ACUF mean, but it's an animal study design. And they want to compare, the PI want to compare the treatment effect between treatment and the placebo using the mice model, because this is animal design study. They have data from 3 mice, and they want to see how many mice they need in order to do compare the tumor size. They're specifically interested in tumor size over time, not any specific time point. They want to see the trend or between the treatment groups. So the usual way is just do 2-sample t-test and do the baseline and one of the time points, later time points. But I managed to do linear mixed model simulations for the sample size power in this specific study because they are interested in the longitudinal change over time. So this is very specific needs from the doctor. It's science related. So we have to provide services that tailor to that specific needs. In this case, we need a linear mixed model instead of 2-sample t-test. And this is, they're really happy with the design advanced design with the simulation results. And this is kind of good positive feedback for me to keep doing this to kind of customize design for each client.
Austin, can I interrupt you for just a second?
Yes.
Yeah, so just for everybody's information, the ACUF is Animal Care and Use Form.
Yes, thank you.
And I think what I wanted to emphasize is you talked a lot about the actual scientific design, but what's another part of an important feature of the Animal Care and Use Form is additional details that the I would say clinicians, the scientists, because they might not be clinicians, would contribute to make sure that the animals are treated fairly, right? Because there's a lot of other things associated with, we could do any kind of study that we wanted to do, right? But we want to do things that are ethically appropriate. And so that's why it's called Animal Care and Use. Anyway, carry on. Just for everybody else's information, that might be something that they're not familiar with. So I thought I'd jump in and share a point.
Thanks. Thank you. That's really, I should do more homework before doing this. I just put ACUF without thinking I wanted to.
But I think it might help others to understand, a little bit more.
Definitely.
Carry on. Thank you.
Okay. So now we are, now we'll get to the actual clinical trial designs. So these are the designs we most often use in our daily work. The phase 1 is dose finding, used to use 3+3 a lot. And still clinicians want to use 3+3 because of simplicity. But now we're using Bayesian design more and more often because it has the same simplicity with predefined rules. And then you can just follow the rules and do the dose escalation, de-escalation. And for phase 2, we usually have Simon's two-stage design, the Bayesian monitoring for toxicity and futility. And for phase 3, for me, I don't have much phase 3 designs because it's usually a large scale study. And I did a couple of them, most of them group sequential design with sample size power calculation, like interim look for both futility and the sensitivity, the efficacy. And we also have sometimes do seamless design, phase 1, 2, platform design with multiple indication, multiple treatment in the same kind of protocol, covered everything. And this is a trial design web page that's provided by MD Anderson Stat Department, Dr. Lee and Dr. Yuan. And this is very, very useful. And I go to here almost all the time. Like here they have Boeing suite, original Boeing, the backfill Boeing, laid on set, and combinations. There's so many designs here. I definitely haven't tried all of them. Boeing design is the one I use the most right now. And Simon's two-stage, of course, for stage 2. And then have Bayesian efficacy monitoring and futility monitoring on this too. So you can go there and check out these designs. They're wonderful and very useful. Okay. All right. So yeah, we need to understand the designs, when to use them and how to use them for sure. And we also need to, when we discuss with the clients what design to use, we have to get their input. So it's also communication is very important because we need to solicit lots of information from doctors. What endpoint, what time frame, what design, what kind of power and sample size limit they can do. A lot of input, lots of communications here. Just personally, I would advise avoid designs that's you have to decide on the go without pre-specified rules. That's a nightmare to run. Just try to avoid those kind of designs. And then also a big chunk of our daily job is to do data analysis. So here I list all the kind of usual or routine analysis we do as a statistician, biostatistician. Survival for sure, for cancer, logistic regression for response, linear model, linear mixed model with longitudinal data. And here, one big topic is model and variable selection. I found it more and more nowadays. I ask the clinicians what they want in the model, because a model is more clinical relevant than is useful than statistical useful. So you have to make sure the model makes sense to the clinicians. So I asked them to pick some variables. We run some different combinations, but pick the one that makes the most sense to them. Again, here we need a lot of input. It's not just statistics. It's clinical and data-driven model fitting. That's the most kind of time-consuming part. You have to make sure your model makes sense and you can interpret the results to the clinicians and to the audience. So talking about designs, we design clinical trials, for example, based on response rate or based on overall survival, progression-free survival. And then we run a trial, and some succeed, some will fail. I know one case that one patient with metastases, stage 4, triple-negative breast cancer, was effectively cured by one treatment. However, that treatment failed the phase 3 trial. So it's like, we know it's a probability of success, but down to one patient, specific patient, that patient was totally cured by the treatment. However, the treatment didn't work on most or enough patients to be successful in phase 3 trial. And how to define success and how to define a treatment benefit, that's a hard question to answer. I don't think there's definitive answer to that. It's so heterogeneous in patient, and it's so hard to find the best choice of endpoints and target for clinical trial designs. I think, to me, that's a limitation we have to keep in mind, that we are designing trials with probabilities, hopefully with higher probabilities to succeed, but still some patient, maybe some subgroup patient may benefit while most of the other patients won't. It's a research or statistical question to find ways to identify the subgroups that can benefit. And that leads to kind of continued education. We have to keep learning. Like after 20 years, I still keep learning new stuff. For example, the phase 1 design, the Boeing design, may not have the backfill option. So you can, instead of finding one dose from phase 1, you find 2 or even more doses after phase 1. It's much more efficient, and they also comply with the Project Optimus, the new policy FDA put in place in 2021. And they want to randomize dose comparisons to find best dose, the balanced dose with efficacy and toxicity before going into large-scale phase 3 trial. So they want model-informed drug development, and they also want to take into considerations the patient report outcomes, like questionnaires. And also they want suggest seamless phase 1/2 study design with dose expansion to find optimal dose. So these are all new things to me, and I keep learning these new policies and design methods. As for software, ggplot2 is kind of lifesaver for me. I remember when I started, I have to write everything myself for plotting. It's hard to read even to myself. And then it's hard to maintain because it's hard to understand. But with ggplot2, it's so easy and intuitive. And you just keep adding components, keep adding modules to what you have right now on top of each other and get what you want with pretty figures. And data visualization is very, very important. I can't emphasize this more. A picture worth 1,000 words for sure. And also AIs, ChatGPT, DeepSeek. Nowadays, I used to kind of search online, Google, what kind of example code or template we can use. Now I just ask ChatGPT or DeepSeek, provide me a chunk of code to do what kind of specific task. And all I need to do is just run it, just make sure it works on the data. And it's so much more efficient than Googling right now. But of course, all the results from ChatGPT or DeepSeek, you have to validate, make sure it works and make sure it's true, because they pretend to know everything and they give you an answer no matter what. If you ask a question, they give you an answer, but you have to make sure the answer is correct. So this is kind of keep learning the new method or new tools, kind of expanding the toolbox over time, even after 20 years for me. So this is Foundation, a tool that Telperian developed for data visualization and some standard service, and also in innovative machine learning capabilities. So I'll show this quickly, kind of go through it. This is a demo. What they do is for Explore, we have a data set here, study overview. This is list all the ADAM data set. We can also do STDM and CSV files. So list all the files in this study, a total of about 15 study here. And if you go to subject level, and you can see the actual data. I used to use R and open like view data, but now this is the actual raw data of the subject level data. And you can see variables. For example, age, you can see distribution by arm, and you can pick or remove arms here, and here's this position plot of the patient. The powerful tool I found it very useful is when you have no ADAM files, very long files, that you stack everything up. It's the longest format. It's really hard to explore when it's in raw format with so many, like for example, this one, questionnaire, and everything
Auston?
is, yes.
Austin, can I interrupt? Someone
Yeah.
is asking, are we able to view this on our own device? I'm not sure if that point can be made clearer. I don't know if you're just,
Well, this one.
is that something you're just showing like in your back? I mean, are you just flipping through a website, or are these actually part of your slides?
Oh, this is a website, sorry.
Okay.
This is I clicked the hyperlink to a website. This is the
Yeah.
foundation
So I guess
website, sorry.
so maybe, I don't know if that website is available publicly, but maybe afterwards you can provide that so they could, if it is.
It's not, I don't think it's publicly available. It's a tool that Talperian built, and and you need a subscription. But I don't know if they have a like demo part that's open public. I'll ask them.
Okay.
Okay. So this is the longitudinal data. You can see that variables here, for example, variable list. The AVL has everything in it with 100,000 lines of data. But with this foundation, you can clearly see it over time, how the data distribute over time. Like for example, here, the baseline, week 24, week 48. It can easily visualize the data and also give you a waterfall plot. Change, you can say percent change, baseline, and change from baseline to absolute change, the percentage. So you can pick whatever, as long as it's reported in the data set, you can see it. And that's, to me, that's very powerful tool to use to actually see the data. And then you decide, because when we do data modeling analysis, we need to know, like I'm a big fan of Dr. Frank Harrell's modeling strategy book. And we want to see the data first and decide how much degree of freedom, how many degree of freedom we want to assign to that variable in the modeling. And this is the great way to actually see the data, explore it. And then we also do analysis. There's so a lot of results. I'll show like correlation. And then associations, endpoints analysis. Like here, we want to compare treatment. And you can see placebo and treatment. And this is interactive. You can put it on any spot. It will show you which patient it is and which group it is and distribution of the whole endpoint, and also compare it with p-value is 0.28. Basically give you the kind of routine biostat, everything in it. All the summaries, table ones, and comparisons. And here's a cool thing I want to show. That's the model, machine learning part. So Brian Hobbs, that's my former colleague, and he's the founder and the chief scientific officer of Talperian. He developed this network kind of clustering, semi-parametric way to cluster patients, identify subgroups that can benefit from treatment. So this is machine learning results of the clustering, and you can see it. We have 4 different groups and can be separated by these 3 variables: region, the baseline cirrhosis status, and also the baseline HVPG data. And you can clearly see this part is clear. Group C is different from others, but ABD kind of mixed together. And if this is confidence based on the kind of distance measure. And if you pull the confidence, I want to see, you only see the dots with 90% or 80% confidence. And then those below 80% will be grayed out, and you left only the patient with higher confidence. And then you can check what their distribution and what their outcome by each subgroup by treatment in this view. So this is very cool. And the last thing I want to show is the patient profile. And sometimes we want to focus on each specific patient, how they kind of progress and how they fare on the trial. And this is an example of one patient, patient one. It gives you the AEs over time. This patient had 2, didn't have CONMAT, and the 2 endpoints, ALT and AST lab values over time, and dashed line are the group means of the treatment group. This is a patient specific, that specific patient values over time. And also the age, gender, race, and all the demographic. And this patient had 5 AEs on trial observed from this day 82 up to day 400. And again, this is patient level data, and dashed line is the group level, group means. Here's another patient. So this is also very useful when we do go over patient and see specifically each patient, how they go, how they did on trial. So these are the, I think that's pretty much all I want to share today. And to me, I think as a master level statistician, I provide routine analysis, biostat or consulting service independently, because with extensive experience in cancer, especially in cancer, I can work with doctors and answer questions and help them with design, data analysis, everything by myself. But for research, new methodologies or new kind of data come in, I definitely need a PhD statistician help kind of get started and to learn the new methodology and also to use it. Because as a master level statistician, I think the most important thing is to how to use it efficiently and how to answer the question, clinical questions, communicate with clinicians, and make sure the methods are not just papers. They are useful. They can be applied to real clinical trials and to real data analysis. And my suggestion is, if you could, get a PhD. I tried, I failed. I couldn't do it with 3 young kids and full-time job and 4 hours round trip to A&M. But I definitely encourage my friends and team members to get a PhD if they can manage it. It's a really big task. All right, I want to thank the PhD collaborators or professors I've worked with in my career. Dr. Ken Hess, I had my first paper with him. Now I have like 200 more, 200 plus papers, but the first one is with Dr. S. Jeff Morris, he's also an Aggie. He came early, in my early career, I worked with Jeff and Val Johnson a lot. Val was the department chair. And we stood to support imaging departments and leukemia departments. Zdine Phone is also helped support the imaging department. Brian Hobbs, we support the whole division of the imaging and also radiation oncology. Sujata Patel is my current PhD supervisor at CCF, and she's very supportive. And when I kind of switched from full-time to part-time, and she's very, very supportive. Also want to thank colleagues and collaborators. Last but definitely not least, Marcy, thanks for inviting. And also, I remember when back to interview, we went to lunch together, right? He asked me why you want to come to MD Anderson. And I said, I want to help cancer patients, and because my grandma died of cancer. And the closest spot I can get is a cancer biostatistician to kind of stand behind or beside the doctors to help cancer patients. And that's still my passion and my dedication. Thanks. That's it.
Thank you, Austin. Yeah, so that, I guess that was just a, like a, snapshot view into your career.
Right.
Beginning from MD Anderson shortly after you graduated from Texas A&M University up until now. I'm sure there's some things that I recognized along the way in your talk that, perhaps maybe some of the participants today might not be familiar with in terms of study designs and reasons why you do some of those things, but hopefully it gives them just some ideas of some of the things that you got to work on and
Right.
from my perspective and how you can contribute back to the community. I think that's one of the benefits of this webinar type series that we have, is to provide information for our students and our former students to better understand what kinds of careers people have, what kinds of things they do on a day-to-day basis, and what kinds
Right.
of contributions that they make to the scientific and even, I guess, different kinds of communities. So I wanted to ask if there's anybody in the audience today that has any questions. If so, please either raise your hand or speak. I think Austin's here for a little while longer, and again, we'd be happy to entertain some questions if anybody has any. Sorry, I'm trying to keep up with the messages here.
Yeah, I'm reading the messages. With single cell or bulk sampling, well,
I think that's when you were
single-
given that
yeah.
presentation of that software.
Oh, okay. Well, this is kind of the demo was based on clinical trials patient, and it has like 500 patients in that data, and that's the data I usually work with, the clinical data from clinical trials, from the retrospective studies. I don't have much work from basic science lab anymore. I used to do some basic science research, like high dimensional data analysis, bioinfo type of analysis, but not anymore.
Yeah, someone just asked a question. Joe Sweet asked a question about, in the clustering you showed, was the left versus right on the 3D visual.
Left versus right. I don't know. Let me. Can I go back to it? Did I close it? Okay, let me try. Go back to. It's not working. Oh, no. All right. So. Oh, do you mean here, left and right? No, it's not. They happen to be kind of two larger clusters. These three cluster together. This three larger cluster together. But it's not a left and right per se. It just subgroup C is more like itself, especially this cluster. And the other ones are more kind of tangled, but somehow they separated here with some separation, maybe because of the baseline HVPG factor or cirrhosis status. We're trying to identify a subgroup that can benefit from the treatment. What I didn't show is a counterfactual analysis that kind of give the counterfactual effect the patient get drug A or placebo in all subgroups and do the comparisons. I didn't show this because I'm not really good at understanding how to explain it. But we can do counterfactual analysis using this foundation tool to identify the subgroups that benefit the most from treatment. Did that answer the question? Sorry.
Yes, he said yes. Thank you.
Okay, great. Thanks.
Yeah.
That's
Any
so
other
cool with the
Oh, I'm
3D.
sorry.
No, I'm
Yeah,
sorry.
it looks very cool.
Yeah.
Yeah. Anyone else? Any other questions, I guess, pertaining to the talk or perhaps otherwise that we might be able to address in today's time? Thank you.
Oh, the name of software is the Foundation.
But you said it's not publicly available, right?
Right. It's, the foundation service itself is not publicly available. You need a prescription to the service. But I don't know about the demo part. Maybe the demo part is public. I'll ask and share information. It's called Foundation.
Someone asked, what do you think helped get you into MD Anderson? Internships, education projects, interview?
Well,
you don't have, does
Texas
it make you uncomfortable to answer that?
No, I think, well, because MD Anderson is my first job, first ever. And I guess the stat department, the education provided by stat department is enough.
Yeah, it's very true. I think that the strength of getting your degree from Texas A&M University is very important. There are many other academic institutions that don't have that same weight, so to speak. So I think that was definitely important. You know, and you know, just because I've interviewed for many years, I think there's no single one of these things is going to be a determining factor. But I think obviously, you know, sometimes it's having the academic training is important, but also having some of the soft skills that are not as easily learned in an educational environment are important as well. So it's I appreciate the question because I think it's a good question, but it's a little hard to answer. New message. Okay. Anyone else? Any other questions or perhaps even comments? Sorry, I guess my video. Back on. Anything else? Anyone?
Going once, going twice.
Yeah, exactly. Exactly. Yeah.
Deal.
We've lost a few people anyway. So,
Okay.
well, I appreciate everybody joining us today. I think Auston gave us some insight into life as a biostatistician for over a couple decades, I think. Give you some different ideas of things that you get to work on, different kinds of folks you get to work with. I don't know how many publications Auston has at this point, but he had, I don't know, many whenever he was working with us at MD Anderson. So it's really valuable work that he's contributed to over the years, and I'm sure continues to be the case. So I encourage you all to keep that in mind if you're job searching.
Yeah.
Although I'm not hiring, but I'm sure that there's somebody who is. So, well, again, thanks everybody for joining us today. This, like I said, this was our March version of the Statistics Former Students Network webinar. If anybody is interested in speaking at a future webinar, just please reach out to me. I'd be happy to take volunteers for more talks. It's always useful information, I think, across the community to see what people are doing. And thanks for joining us. And thanks, Austin, for sharing with us today.
Thank you.
Everybody have a great day. Take care.
Okay.